11 Chapter 13 Plant Growth and Development. You have already studied the organization of a flowering plant in Chapter 5. Have you ever thought about where and how the structure like roots, stems, leaf, flower, fruit, and seed arise, and that too in an orderly sequence? You are by now aware of the terms seed, seedling, plantlets, mature plants. You have also seen that trees continue to increase in height or breadth over a period of time. However, the leaves, flowers, and fruits of the same tree are not only have limited dimension but also appears and fall periodically and sometimes repeatedly. Why does vegetative phase proceed flowering in a plant? All plant organs are made up of variety of tissue. Is there any relationship between the structure of a cell, a tissue, an organ, and the function they perform? Can the structure and the function of these be altered? All cells of a plant are descendants of the zygote. The question is then why and how do they have different structure and function attributes? Development is the sum of two processes, growth and differentiation. To begin with, it is essential to sufficient to know that the development of a mature plant from a zygote fertilized egg follow a precise and highly ordered succession of events. During this process, a complex body organization is formed that produce roots, leaves, branches, flowers, fruit, and seeds, and eventually they die. Figure 13.1. The first step in the process of plant growth is seed germination. The seed germination, when favorable conditions for growth exist in the environment. In the absence of such favorable conditions, the seeds do not germinate and goes on into the period of suspending growth or rest. Once favorable conditions return, the seed resume metabolic activities and growth take place. In this chapter, you shall also study some of the factors which govern and control these developmental processes. These factors are both intrinsic, internal, and extrinsic, external to the plant. Figure 13.1 Germination and seedling development in peas. Pea. 13.1 Growth. Growth is regarded as one of the most fundamental and conspicuous characteristic of a living being. What is growth? Growth can be defined as an irreversible permanent increase in size of an organ or its part or even of an individual cell. Generally, growth is accompanied by metabolic processes, both anabolic and catabolic, that occur at the expense of energy. Therefore, for example, expansion of a leaf is growth. How would you describe the swelling of piece of wood when placed in water? 13.1.1 Plant growth generally is indeterminate. Plant growth is unique because plants retain the capacity for unlimited growth throughout their life. This ability of the plant is due to the presence of meristems at certain locations in their body. The cells of such meristem have the capacity to divide and self-perpetuate. The product, however, soon lost the capacity to divide, and such cells make up the plant body. This form of growth, wherein new cells are always being added to the plant body by the activity of the meristem, is called the open form of growth. What would happen if the meristem ceases to divide? Does this ever happen? In earlier classes, you have studied about the root apical meristem and shoot apical meristem. You know that they are responsible for the primary growth of the plant and principally contribute to the elongation of plant along their axis. You know that in dicotyledonous plant and gymnosperms, the lateral meristem, vascular cambium, and cork cambium appear later in life. These are the meristems that cause the increase in the growth of the organ in which they are active. This is known as secondary growth of the plant. See figure 13.2. Figure 13.2 Diagrammatic representation of location of root apical meristem, shoot apical meristem, and vascular cambium. Arrows exhibit the direction of growth of cells and organs. 13.1.2 Growth is measurable. Growth at a cellular level is principally a consequence of increase in the amount of protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measure some quantity which is more or less proportional to it. Growth is therefore measured by a variety of parameters, some of which are increase in fresh weight, dry weight, length, area, volume, and cell number. You may find it amazing to know that one single maize root apical meristem can give rise to more than 17,500 new cells per hour, whereas cell in a watermelon may increase in size by up to 350,000 times. In the former, growth is expressed as increase in cell number. The latter expresses growth as increase in size of the cell, while the growth of a pollen tube is measured in terms of its length. An increase in surface area denotes the growth of in a dorsiventral leaf. 13.1.3 Phase of growth. The period of growth is generally divided into three phases, namely meristematic, elongation, and maturation. Figure 13.3. Let us understand this by looking at the root tips. The constantly dividing cell both at the root apex and the shoot apex represent the meristematic phase of growth. The cells in the region are rich in protoplasm, possesses large conspicuous nuclei. Their cell walls are primary in nature, thin and cellulosic with abundant plasmodesmatal connection. The cells proximal just next away from the tip to the meristematic zone represent the phase of elongation. Increased vacuolation, cell enlargement, and new cell wall deposition are the characteristics of the cells in the phase. Further away from the apex, more proximal to the phase of elongation, lies the portion of axis which is undergoing the phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening and protoplasmic modification. Most of the tissues and cell types you have studied in earlier classes represent this phase. 13.1.4 Growth rates. The increased growth per unit time is termed as growth rate. Thus, rate of growth can be expressed mathematically. An organism or a part of the organism can reproduce more cells in a variety of ways. Figure 13.4 Diagrammatic representation. A. Arithmetic, B. Geometric growth, and C. Stages during embryonic development showing geometric and arithmetic phases. The growth rates show an increase that may be arithmetic or geometrical. Figure 13.4 An arithmetic growth following mitotic cell division, only one daughter cell continues to divide while the other differentiates and matures. The simplest expression of arithmetic growth is exemplified by a root elongation at a constant rate. Look at figure 13.5. On plotting the length of the organ against time, a linear curve is obtained mathematically. It is expressed as LT is equal to LO dot plus RT. LT is equals to length at time t. LO is equals to length at time 0. R is equals to growth rate or elongation per unit time. Let us now see what happens in geometrical growth. In most systems, the initial growth is slow lag phase and it increases rapidly thereafter at an exponential rate, log or exponential phase. Here both the progeny cells following mitotic cell division retain the ability to divide and continue to do so. However, when limited nutrient supply, the growth slows down, leading to a stationary phase. If we plot the parameter of growth against time, we get a typical sigmoid or S curve. Figure 13.6. A sigmoid curve is a characteristic of living organism growing in a natural environment. It is typical for all cells, tissue, and organs of a plant. Can you think of more similar example? What kind of a curve can you expect in a tree showing seasonal activities? The exponential growth can be expressed as W1 is equals to W0 ERT. W1 is equals to final size, weight, height, number, etc. W0 is equals to initial size at beginning of the period. R is equals to growth rate. T is equals to time of growth. E is equals to base of natural logarithm. Here R is the relative growth rate and is also the measure of the ability of the plant to produce new plant material, referred to as efficiency index. Hence, the final size of W1 depends on the initial size W0. Quantitative comparison between the growth of living system can also be made in two ways. First, measurement and the comparison of total growth per unit time is called the absolute growth rate. Second, the growth of the given system per unit time expressed on a common basis. Example, unit initial parameter is called the relative growth rate. In figure 13.7, two leaves A and B are drawn that are of different sizes but shows absolute increase in area in the given time to give leaves A1 and B1. However, one of them shows much higher relative growth rate. Which one and why? 13.1.5 conditions for growth. Why do you not try to write down what you think are necessary conditions for growth? This list may have water, oxygen, and nutrients as very essential elements for growth. The plant cell grow in size by cell enlargement, which in turn require water. Trajectory of cell helps in extension growth. Thus, plant growth and further development is intimately linked to the water status of the plant. Water also provides the medium for enzymatic activities needed for growth. Oxygen helps in releasing metabolic activities essential for growth activities. Nutrient, macro and micro essential elements are required by plant for the synthesis of protoplasm and act as source of energy. In addition, every plant organism has an optimum temperature range best suited for its growth. Any deviation from the range could be detrimental to its survival. Environmental signals such as light and gravity also affect certain phases or stages of growth. 13.2 Differentiation, Dedifferentiation and Redifferentiation. The cells derived from root apical and shoot apical meristem and cambium differentiate and mature to perform specific function. This act leading to maturation is termed as dedifferentiation. During differentiation, cells undergo few to major structure changes both in their cell wall and protoplasm. For example, to form a tertiary element, the cell would lose their protoplasm. They also develop a very strong elastic lignocellulosic secondary cell wall to carry water to long distance even under extreme tension. Try to correlate the various anatomical features you encounter in plant to the functions they perform. Plants show another interesting phenomenon: the living differentiation cells that by now have lost the capacity to divide and can regain the capacity of division under certain conditions. This phenomenon is termed as dedifferentiation. For example, formation of meristem, interfascicular cambium and core cambium form fully differentiated parenchyma cell. While doing so, such meristem or tissue are able to divide and produce cells that once again lose the capacity to divide but mature to perform specific function. Get redifferentiation. List some of the tissue in a woody dicotyledonous plant that are products of redifferentiation. How would you describe a tumor? What would you call the parenchyma cells that are made to divide under controlled laboratory conditions during plant tissue culture? Recall in section 13.1.1 we have mentioned that the growth in plants is open. It can be indeterminate or determinate. Now we may say that even dedifferentiation in plant is open because cells or tissue arising out of the same meristem have different structure at maturity. The final structure at maturity of a cell or tissue is also determined by the location of the cell within. For example, cell position away from root apical meristems differentiates as root cap cells, while those pushed to the periphery mature as epidermis. Can you add a few more examples of the open differentiation correlating the position of a cell to its position in an organ? 13.3 Development. Development is a term that includes all changes that an organism goes through during its life cycle from germination of the seed to senescence. Diagrammatic representation of the sequence of processes which constitute the development of a cell of a higher plant is given in figure 13.8. It is also applicable to tissue or organ. Figure 13.8 Sequence of a developmental process in a plant cell. Plant follow different pathway in response to environment or phases of life to form different kinds of structure. This ability is called plasticity. Example: heterophyll in cotton, coriander and larkspur. In such plants, the leaves of the juvenile plant are different in shape from those in mature plants. On the other hand, difference in shapes of leaf produced in air and those produced in water in buttercup also represent the heterophilous development due to environment. Figure 13.9. This phenomenon of heterophily is an example of plasticity. Figure 13.9. Heterophily in A. Larkspur, B. Buttercup. Thus, growth, differentiation, and development are very closely related events in the life of a plant. Broadly, development is considered as the sum of growth and differentiation. Development in plant, both growth and differentiation, is under control of intrinsic and extrinsic factors. The former include both intracellular genetic or intracellular factor chemicals such as plant growth regulator, while the later include light, temperature, water, oxygen, nutrition, etc. 13.4 Plant growth regulator (PGR). 13.4.1 Characteristics. The plant growth regulator (PGRs) are small, simple molecules of diverse chemical composition. They could be indole compounds, indole 3 acetic acid IAA, adenine derivatives N6, furfuryl amino, purine, kinetin, derivatives of carotenoid, abscisic acid ABA, terpenes, gibberellic acid GA3 or gases, ethylene C2H4. Plant growth regulator are variously described as plant growth substance, plant hormone, or phytohormones in literature. The PGR can be broadly divided into two groups based on the function in a living plant body. One group of PGR are involved in growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. These are also called plant growth promoter. Example: auxin, gibberellin, and cytokinin. The PGR of the other group play an important role in the plant response to wound and stress of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscission. The PGR abscisic acid belong to this group. The gaseous PGR ethylene could fit either of the groups, but it is largely an inhibitor of growth activities. 13.4.2 The discovery of plant growth regulator. Interestingly, the discovery of each of the 5 major groups of PGR have been accidentally. Started with the observation of Charles Darwin and his son Francis Darwin, when they observed that the coleoptiles of canary grass respond to unilateral illumination by growing towards the light source, phototropism. After a series of experiments, it was concluded that the tip of coleoptile was the site of transmittable influence that caused the bending of the entire coleoptile. Figure 13.10 Auxin was isolated by FW went from tips of coleoptile of oat seedling. Foolish seedling disease of rice seedling was caused by a fungal pathogen, Gibberella fijicurea. E. Kurosawa, 1926, reported the appearance of the symptoms of the disease in rice seedling when they were treated with sterile filtrates of the fungus. The active substance were later identified as gibberellic acid. F. Scotch and his co-worker observed that from the internodal segment of tobacco stems, the callus, a mass of undifferentiated cell, proliferated only if, in addition to auxin, the nutrients medium was supplemented with one of the following extracts of vascular tissue: yeast extract, coconut milk, or DNA. Miller et al. 1955 later identified and crystallized the cytokinesis promoting active substance that they termed kinetin. During mid-1963, independent researchers reported the purification and chemical characterization of 3 different kinds of inhibitor B, incision second, and dormin. Later, all the 3 were proved to be chemically identified. It was named abscisic acid ABA. H.H. Cauchin, 1910, confirmed the release of the volatile substance from ripened organ that hastened the ripening of storage unripened bananas. Later, this volatile substance was identified as ethylene, a gaseous PGR. Let us study some of the physiological effects of these 5 categories of PGR in the next session. 13.4.3 Physiological effect of PGR. 13.4.3.1 Oxen. Oxen from Greek oxen to grow was first isolated from human urine. The term oxen is applied to the idol 3-acetic acid IAA and to other natural and synthetic compounds having certain growth regulator properties. They are generally produced by the growing apex of the stem and roots from where they migrate to the region of their action. Oxen like IAA and idol butyric acid IBA have been isolated from plants NAA, naphthalene, acetic acid, and 2,4-D, 2,4-dichlorophenoxyacetic are synthetic oxens. All these oxens have been used extensively in agricultural and horticultural practices. They help to initiate rooting in stem cutting, an application widely used for plant propagation. Oxen promote flowering. Example, in pineapples, they help to prevent fruit and leaf drop at early stage but promote the abscission of older mature leaves and fruit. In most higher plants, the growing apical bud inhibits the growth of the lateral auxiliary buds, a phenomenon called apical dominance. Removal of shoot tips deception usually result in the growth of laterally buds. Figure 13.11. It is widely applied in tea plantation hedge making. Can you explain why? Oxen also induce parthenocarpy. Example, in tomatoes, they are widely used as herbicides. 2,4-D, widely used to kill dicotyledonous weed, does not affect mature monocotyledonous plant. It is used to prepare weed-free lawns by gardeners. Oxen also controls xylem differentiation and helps in cell division. 13.4.3.2 Gibberellin. Gibberellin are another kind of promotory PGR. There are more than 100 gibberellin reported from widely different organisms such as fungi and higher plant. They are denoted as GA1, GA2, GA3 and so on. However, gibberellic acid GA3 was one of the first gibberellin to be discovered and remain the most intensively studied form. All GAs are acidic. They produce a wide range of physiological response in the plant. Their ability to cause an increase in length of axis is used to increase the length of grape stalks. Gibberellin cause fruit like apple to elongate and improve its shape. They also delay senses, thus the fruit can be left on the tree longer as to extend the market period. GA3 is used to speed up the malting process in brewing industry. Sugarcane store carbohydrate as sugar in their stem. Spraying sugarcane crop with gibberellin increase the length of the stem, thus increase the yield by as much as 20 tons per acre. Spraying juvenile conifers with GA hastens the maturity period, thus leading to early seed production. Gibberellin also promotes bolting internode elongation just prior to flowering in beet, cabbage, and many plants with rosette habitats. 13.4.3.3 Cytokinins. Cytokinins have specific effect on cytokinesis and were discovered as kinetin, a modified form of adenine and purine from entoclaved herring sperm's DNA. Kinetic does not occur naturally in plants. Search for natural substances with cytokinin-like activities lead to the isolation of zeeth and corn kernels and coconut milk. Since the discovery of zeatin, several naturally occurring cytokinesis and some synthetic compound with cell division promoting activity have been identified. Natural cytokinesis are synthesized in region where rapid cell division occur, for example root, apex, developing shoot buds, young fruit, etc. It helps to produce new leaves, chloroplast in leaves, lateral shoot growth and adventitious shoot formation. Cytokinin helps overcome the apical dominance. They promote nutrients mobilization which helps in the delay of leaf synthesis. 13.4.3.4 Ethylene. Ethylene is a simple gaseous PGR. It is synthesized in a large amount by tissue undergoing synthesis and ripening fruits. Influence of ethylene on plant include horizontal growth of seedling, swelling of axis and apical hook formation. Dicot seedling. Ethylene promote synthesis and abscission of plant organs, especially of leaves and flowers. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of the fruit. This rise in rate of the respiration called the respiratory, climatic. Ethylene breaks seeds and bud dormancy, inhibit germination in peanut seeds, sprouting of potato tuber. Ethylene promote rapid internode petiole elongation in deep water rice plant. It helps leaves upper parts of the shoot to remain above water. Ethylene also promotes root growth and growth hair formation, thus helping the plant to increase their absorption surface. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapples. It also induces flowering in mangoes. Since ethylene regulates so many physiological processes, it is one of the most widely used PGR in agriculture. The most widely used compound as source of ethylene is ethiphenone. Ethiphenone is an aqueous solution, is readily absorbed and transported within the plant and release ethylene slowly. Ethiphenone hastens fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits. Thinning of cotton, cherry, walnut promote female flower in cucumber, thereby increasing the yield. 13.4.3.5 Abscisic acid. As mentioned earlier, abscisic acid ABA was discovered for its role in regulating abscission and dormancy, but like other PGR, it also has the other wide-ranging effects on plant growth and development. It acts as a general plant growth inhibitor and an inhibitor of plant metabolism. ABA inhibitor seed germination. ABA stimulates the closure of stomata and increase the tolerance of plant to various kinds of stress. Therefore, it is called the stress hormone. ABA plays an important in seed development, maturation, and dormancy. By inducing dormancy, ABA helps seed to withstand desiccation and other factors unfavorable for growth. In most situations, ABA acts as antagonist to GAs. We may summarize that for any and every phase of growth, differentiate, and development of plant, one of the other PGR has many roles to play. Such roles could be complementary or antagonist. These could be individualistic or synergistic. Similarly, there are a number of events in the life of a plant where more than one PGR interact to affect that event. Example: dormancy in seed, bud, abscission, sensation, apical dominance, etc. Remember, the role of PGR is of only one kind of intrinsic control. Along with the genomic control and extrinsic factor, they play an important role in plant growth and development. Many of the extrinsic factors such as temperature and light control plant growth and development via PGR. Some of such events could be vernalization, flowering, dormancy, seed germination, plant movement, etc. We shall discuss briefly the role of light and temperature, both of them the extrinsic factor on initiation of flowering. Summary: growth is one of the most suspicious events in any living organisms. It is an irreversible increase expressed in parameters such as size, area, length, height, volume, cell number, etc. It conspicuously involved increased protoplasmic materia in plant meristema are the site of growth. Root and shoot apical meristem, sometimes along with the intercalary meristem, contribute to the elongation growth of plant axis. Growth is indeterminate in higher plant following cell division in root and shoot apical meristem cell. The growth can be arithmetic or geometrical. Growth may be not and generally is not sustained at a high rate throughout the life of a cell, tissue, organ, organism. One can define three principal phases of growth: the lag, the log, and senescent phase. When a cell loses the capacity to divide, it leads to differentiation. Differentiation results in development of structure that is commensurate with the function. The cell finally has to perform. General principles for differentiation for cells and tissue and organs are similar. A differentiation cell may de-differentiate and then re-differentiates, and differentiation in plant is open. The development could also be flexible. The development is the sum of growth and differentiation. Plant exhibit plasticity in development. Plant growth and development are under the control of both intrinsic and extrinsic factor. Intercellular intrinsic factor are the chemical substance called plant growth regulator PGR. These are diverse group of PGR in plants, principally belonging to 5 group: auxin, gibberellin, cytokinin, abscisic acid, and ethylene. These PGR are synthesized in various parts of the plant. They control different differentiation, and developmental events. Any PGR has diverse physiological effect on plant. Diverse PGR are also manifest similar effects. PGR may act synergistically or antagonistically. Plant growth and development is also affected by plant temperature, nutrition, oxygen status, gravity, and such external factors.